Good morning, folks. This is Mr. Dan coming to you from the Landscape Design Classroom, and today I am here to talk to you about the propagation of plants. So, two ways plants are reproduced, from seed, sexual reproduction, and through vegetative propagation, that's asexual reproduction. So today we're going to focus on this asexual form of reproduction. So, sexual reproduction in plants requires flower formation, pollinization and fertilization, seed development, and seeds grow into new plants. So pollination, the transfer of pollen from anther to stigma, and you can actually see this process down here in the right corner with a, looks like a little bee landing on top of it, take some of that pollen out and move it over to another plant. So self-pollination, pollen is transferred from the anther of a flower to the stigma of the same flower or another flower on the same plant. So cross-pollination, pollen is transferred from the anther of one flower to the stigma of a flower on another plant of the same species. And like I said, you can see that on the picture down here on the right of a bee taking pollen from one and most likely taking it over to another one. So it may happen by wind or through insects. So the function of fruit. So aids in the dispersal of seeds. And some fruits develop a fleshy edible wall that attracts fruit eaters, which will disperse the seeds in their droppings. So the function of fruit continued. Others develop a dry, hard wall that splits open, allowing the seeds to be shaken out by some disturbance. Some, such as the dandelion, develop light, feathery structures that are carried by the wind, and others have hooks or barbs that stick to passing animals. So moving into seeds, the unit of dispersal for a new plant, some protection from injury and drying, some nourishment for the new plant until it can make its own food, and living organisms in a dormant or inactive state. So most seeds have three parts. So you have a seed coat, which protects the seed, a supply of stored food, cotyledons and endosperm, which feed the embryo, and then the embryo itself, which is made of three parts. They include a young shoot, which is plumule, stem, hypocotyl, and the root, which is radical. So what do seeds need to grow? Conditions inside and outside the seed must be suitable. So moisture, oxygen, and temperature are most critical. The seed's viability, its ability to germinate, depends on this kind of seed and storage conditions. So cool, dry conditions allow for best viability. Asexual reproduction, some plants do not produce seeds. Some seeds do not produce plants identical to parents. So asexual reproduction ensures the desired plant used to produce dwarf plants and used to maintain particular specimens. So the 8 common types of asexual reproduction are cuttings, grafting, budding, layering, dividing, rhizomes, stolons, and tillers or suckers. So asexual reproduction into cuttings. So most common method of asexual reproduction, stems, leaves, or roots, which when detached from a parent plant are capable of reproducing plants exactly like the parent. Inserted into rooting medium, and examples are stem cuttings, tip cuttings, medical cuttings, cane cuttings, hill cuttings, leaf cuttings, and root cuttings. So if you see my little picture here, we have some Clonex here, which is actually what is used to dip that, what you're going to dip that in as a rooting medium, which will help it grow. You can get away with it without using a rooting medium, but it might take a little bit longer for that process to occur, and you will have to take extra care. So asexual reproduction, grafting and budding. Grafting and budding join plants together from 2 plants so they will grow as one plant. Commonly used for fruit and nut trees. The scion is a piece of shoot with dormant buds that will produce the stem and branches. The rootstock or stalk provides the new plant's root systems and sometimes the lower part of the stem. And some examples of this are cleft grafting, bark grafting, and whip or tongue grafting. So budding joins one bud and a small piece of bark from the scion with a rootstock. Now moving into layering. This is stems still attached to their parent plants may form roots where they touch a rooting medium. So once severed, the rooted stem becomes a new plant. Layering is enhanced by girdling the stem where it is bent by wounding one side of the stem or by bending it very sharply. So examples of this would be tip layering, simple layering, compound layering, mound stool layering, and air layering. Now, some more types of layering. So the following can be considered types of layering as the new plants form before they are detached from their parent plants. So stolons and runners, offsets, separation, and division. So looking back at separation, you have bulbs or corms. And then moving into micropropagation, this is also called tissue culture. It's been fairly recent in development back in the 1970s and 1980s. Uses very small pieces of plant tissue grown on sterile nutrient medium under aseptic conditions in a small glass container. Those are called explants. So may use entire seeds, shoot tips, pollen grains to regenerate new shoot systems that can be separated for rooting and growing into full-size plants. Some nurseries use this method and requires trained technicians working under sterile conditions. So you have to be very careful whenever you do tissue culture that you don't get anything into it that would cause it to spoil or become not so much hazardous, but ungrowable. So more about micropropagation used for plants such as ferns, orchids, gerberas, carnations, tobacco, chrysanthemums, asparagus, gladiolus, strawberries, and many other herbaceous plants. Eliminates pathogens and maintains germplasm. The nutrient media for growing excised plant parts includes mineral salts, sugar, vitamins, auxins, cytokines, and sometimes organic complexes such as coconut milk, yeast extract, and banana puree. So looking back into propagation, there is a lot to do here, and we will be doing some of this here in class with the exclusion of tissue culture because it is very hard to keep that type of sterile environment. But we will be doing cuttings and layering and whatnot. So I appreciate your time, folks. If you have any questions at all, please contact me down here in the landscape design classroom or shoot me a message here on YouTube. But have a great day, folks, and thanks for listening.